All right, guys, so here is the little video on how to access your DNA information to figure out what is the actual species of fish that you guys took the DNA from. So I'm going to walk you through the steps here. The links are there and all the information is sitting in your Blackboard shell. So the first thing you need to do is go into the Lectures tab, go down to Biotechnology, and then there are a couple documents posted. One document is the Biotech Code document here. You need to know what your sequence, your tubes say. Are you the MHB2 tube, the RKB tube, seq1, etc. You got to know that to then be able to figure out what is your actual code of information. So make sure you know this. And then go to the next document, the gene sequence. This is the information I got back from Sequitech. So what you do here is you find your particular code. So let me find one here for an example. 2SEQ-MHB. So whoever's tube that is. You sent in that tube, and then Sequitech sequenced the DNA. They did a forward and they did a backwards sequencing. So that's why it says forward. That's why it says reverse. Find the one with the longest stretch of genetic information here. So I would choose, for this person or this group, I would choose this one. I want you guys to copy this information. Okay? So make sure you just highlight all of it, copy it. Go back to here, and now go back to announcements, and this is the website you want to go to. So you need to go to this BLAST website. So you can just copy and paste that, or if you can go out and go Google, and you can just Google BLAST, and this is the site you need. BLAST, Basic Local Alignment Search Tool. National database, free, nothing to it. Okay, so now that you're here, you want nucleotideblast. Click on that. Boom. This box here, enter accession number, that is a gene sequence you need to paste. You need to paste it into here. So let's paste in that sequence. There's all the, notice the GATs, oh, there's a couple Cs, etc. That is literally the nucleotide sequence on the DNA strand you sent to them. So how cool is that stuff? So do that, scroll down, blast. It's going to take a few seconds, and what it's doing is aligning your DNA to what's in the database, the known gene sequences in the database. Depending on how good of a sequence you got, you should hopefully get some results. If we didn't get a lot of DNA, there may not be enough to sequence to match up. Just depends. But everybody did a really good job on it, got some really nice samples. Let's see what fish this is. All right, so here's the sequence alignments. Proximus annularis voucher. So you come over here and you go, all right, it's about a 99.53% identity. Okay, so you got this 99.53% matchup of your DNA sample to this DNA sample in the database. And you go down the list and go, okay, 99.53 here, 99. Okay, this is all looking really, really solid. So what is Proximus annularis? I have no clue. So this is where we go here and we copy that, and then we go to our friend Google. Yep, let me get. And what you want to do is just go to Google, paste in that name. That is the scientific name. Genus, species, subspecies. Scientific name for some fish or some species. We're assuming fish. And let's see what it is. Well, looks like Proximus annularis voucher is lining up with white crappie. So we would check the database, go, okay, that fish sequence is of white crappie. So the database, the blasting information tells us whoever had that sample, your sample said your fish is crappie, that white crappie. So was that your hypothesis? Did you hypothesize it was white crappie, or was it possibly something else? So use this, take a look at it, see if your hypothesis panned out. Do you have data to support the hypothesis? Do you have data to reject it? So the more we sequence DNA, the more we can build this database out, and the more knowledge we can gain. All right, so pretty simple. You guys all did the steps, saw how to do it. Now you have some of the basic skills for a biotechnology program.